All right, so I'm really honored to be able to open Tech Tuesday today. My name is Jamie Dwyer, and I am the director of the Utah Data Coordinating Center and also the assistant vice president for clinical research at the University of Utah Health. I'll just say a sentence or two about what the DCC is and what we're trying to build here. But I'm going to do that at the end after I thank our sponsors tonight for Tech Tuesday, Michael Best, Ballard Spar, Barclay Damon, Workman Nidegger, and Thorpe Northwestern. Thanks to them for their sponsorship for this evening's event. I just want to give a very brief overview about what to expect. So we're going to have a pretty casual conversation among these three members here of the Utah ecosystem. And we're going to be really talking about technology commercialization with experts in the field in the industry field. So I think one of the things that's kind of unique about what we're doing here in Utah is we are really trying to blend industry and academia in unique and maybe novel ways. And I think some of the experience that you'll get to hear about today and that I hope becomes a recurring theme will be evident after that conversation. There's going to be a Q&A afterwards. We really want everybody to ask questions, ask hard questions, especially of Wes and Randy and Bob. I'm sneaking out, so you can't ask me any questions.
We have your cell number.
Yeah, it got posted on the research.utah.edu. I'm opening today, but I'm really here just representing a piece of the ecosystem. The Utah DCC is, as I say, an acronym out of nowhere. We are a full-service academic CRO, or an academic research organization that can partner with industry and academic and philanthropic partners to design and execute multi-site research. And we do all of that over there in the Williams building. I really wish I was in this building. This building's far nicer, but also far more expensive. And we have 130 staff and about 20 faculty who can bring your clinical trial to life. That's a GE quote. But that's really what we do. And so at this point, I also want to acknowledge Dr. Amy Goodman, who's here with us on behalf of the Utah DCC and the work that we do. But at this point, I'm going to turn it over to Randy Peterson, who's going to MC for us tonight.
Well, thanks, Jamie, and thanks to all of you for being here. It's really exciting to all be together, and we have two wonderful guests today. Bob Winelek, who is the COO of Dysgenics, and Wes Sundquist, who's a distinguished professor at the University of Utah. So kind of a biotech academia duo here, and I'll be moderating today. And both of these panelists are really rock stars. In fact, Wes is here fresh off being named one of Time's 100 most influential people this year. So, and let's just cut to the chase, Wes. We're dying to know, what's it like to party with Serena and Snoop Dogg?
So I could... Just so much.
I can tell you that my kids don't think I'm one of the most 100 influential people in the world, but they're infinitely amused that I could ever be on a list with Snoop Dogg. Right, so it was an amazing experience because we got to go to walk the red carpet, and I did walk in right behind Snoop Dogg, even if he doesn't know it.
That's awesome. Well, Bob, we need to know which rock stars you're partying with, but more broadly, you're also involved in really a big culture-changing effort. Can you tell us a little bit about how you think about impacting the world with your own work?
Yeah, I mean, a lot of it is with the different directions, therapies, opportunities everyone's looking at in this room is what impact can some of these emerging therapies have on civilization. If you look at some of the disease states right now, I use the analogy of regenerative medicine, which is my area. It's like the tip of an iceberg. There's not a lot of these therapeutics that are approved right now, but under the waterline, there's this big bubble that over the next 15 years is going to come to fruition and make it to the regulatory pathways. I'll set a couple numbers for you. estimates on the regenerative medicine market, so that means cell, gene therapies, and also other types of immunotherapies, is valued at $42 billion. If we look at 2034, the market's expected to grow to $140 billion. So it's a huge impact, not just financially to the system, but really on how it's going to actually modify diseases for patients. So we can have a huge impact.
Thank you. It seems that in the past, the academic scientist was in the lab and the CEO or COO was in the boardroom. Do you feel like it's a different moment in time where it's more important for the COO and the professor to be out in the public space like you two have been?
I mean, it's always about being in the public space. I mean, number one is the patient, right? I mean, that's what we're doing this for. So understanding the needs of the patient is what drives the product. It's what drives the structure of your clinical trials. And with that patient, it's going to be the clinician. So those are the two most important stakeholders that we're driving through, and to make sure we can be good stewards of that, how we navigate with the regulators, how we raise money to fund the mission, and how we execute, to make sure we can stay on track with the milestones towards approval the best we can. How about you, Wes?
Yeah, so I don't have to convince this crowd, but commercialization's always been important for academic departments, but it's more important than ever, I think, for a couple of reasons. One is the challenge of diversifying our funding sources. But the other is, the tools are just so amazing right now that people should be thinking about how does their basic research translate and have an impact. And so I'm happy to answer questions, but our biochemistry and other, College of Pharmacy and other entities at the U are really making a concerted effort to change our culture and think translationally.
That's great. Why don't we just jump into why you're both here? Would you mind each taking a minute or two and just telling us about what propelled you into the innovation space and into thinking about treatments and therapies?
In mine, I've been in the musculoskeletal industry for forever. I was in it from a device surgical implant business for spine, orthopedic surgery. And I basically was thinking, I said, 1,000 years from now, when they're in Nerthic patients and they're finding all this metal rods, metal implants, they're going to think, what type of barbaric torture was being done to people back in 2020, 2025? And then you look at the prospect of things such as in situ tissue engineering, using biomaterials or using cells or other signals to actually regenerate the parts we wear out over the years of playing here in the Wasatch, for example. And that's a lot of it. And, I said there's got to be a better way as well as our associates and the leadership team. So we decided late in our careers, a number of us, to say, hey, let's do a startup based upon a premise of a novel, unique cell that could actually regenerate the disc in the back for chronic back pain and obviate the need for spinal fusion and get people off opioids. So that was really the driving mission behind it. And then, a lot of it is with the ideation, when you see innovation, is to think with the end in mind. So it's obviously the patient, but given the type of pathway, regulatory pathway, am I going to make it myself? Do we want to take it all the way through the approval route? Do we want to license it out? Do we want to do M&A at a certain point? You really have to think with the end in mind and structure a plan that'll help you get there with the right people on the bus.
Great, thank you.
So maybe I'll just talk about what our lab was involved in. So we've been involved in the development, which is a drug which was developed by Gilead Sciences, which prevents HIV transmission for 6 months from a single shot. And we're very excited about that. Maybe I'll just tell you about how our lab started. So we started here in 1992. And like everybody else, I interviewed a bunch of places. And one of the things you do when you interview is you give a chalk talk about your ideas. And by the time I finished interviewing, I decided my ideas were kind of crap. And so fortunately, the University of Utah made me an offer, and I talked to Dana Carroll and asked him if we could take an extra year to think about what I really wanted to do. And 1991 was the year that Magic Johnson declared he was HIV positive, and There were exponential growth of deaths in men in the US. We had no idea of the problem of HIV/AIDS in Africa. And so I've always been interested in how proteins assemble. And you may know that HIV is unusual in that it has a structure called the capsid, which takes the shape of a cone. And this is still the only biological molecular cone that we know about in biology. And so we were really just driven by trying to understand how do proteins assemble to make a cone that has a wide end and a narrow end. But of course, we are also aware of the fact that this was a really important biological problem, medical problem, and if we could make progress, it might have an impact eventually.
Oh, that's great.
I should also say, I meant to say, that this is always done as a team, and so we're in partnership with Chris Hill, who many of you will know. We had amazing trainees, Barbie Pornillis, Owen Pornillis, who have now been recruited back to the U as faculty members. They were graduate students in the lab. Sam Lee was a graduate student, John Finch. And so I feel like as PI, people credit you with the discoveries, but of course, they're the people who actually make the discoveries.
Awesome. Well, I think it's really fantastic that we have some of academia and industry here. I want to talk a little bit about that relationship and not even the handoff, but the partnership that happens. Now, you've had this really beautiful partnership with Gilead, and Bob, I know you've had a big role in connecting with academia. Would you each talk a little bit about how you see that relationship?
Sure. Yeah, so there's a couple different ways it goes. I mean, obviously there's the initial idea or product concept, which can be tech transfer. A lot of it comes from academia in there, and then how you corroborate on that. And then you look at your team, you build, a lot of our initial team, and part of it, came from academia. But also the roles of academic teaching institutions, for partnerships on animal studies, different types of preclinical things, and then involvement in clinical research. It is a big part, for clinical trial sites and having a balance of those sites as well as the investigators. And then certainly, publications, a big part of taking a therapy through the pipeline and being able to support fundraising is having, robust efficacy data. So it's important to have participation with academia, people that can help legitimize the data you're collecting, get proper peer-reviewed publications, and then, that type of involvement is critical. You can't do without it.
Right, so I left you that we were studying the capsid, and the two things we being Barbie Ganser-Pornillis and Uta von Schwedler did were to show that the capsid in HIV was an architectural object called a fullerene cone. And so we now understood how it was organized. And what Uta did was make small mutations in the capsid, and the surprise was that almost every mutation that she made blocked viral replication, which told us that it was a surprisingly important component of the virus. And then, so that took about a decade, from 92 until 2003, Uta's paper was published. And in 2005, Thomas Seelar, who's the VP for virology at Gilead, called us, cold called and said, do you think capsid would make a good drug target? And if you're an academic lab working on capsid and a great drug company like Gilead asks you that question, you say yes. But we really did think that based on Uta's work, that even small changes in the capsid were inactivating for the virus. So Thomas came and visited, and we hit it off immediately. It's to his great credit that he went back to Gilead and convinced them that capsid could be a drug target for reasons we could talk about. There's lots of reasons that it was not a fashionable kind of drug target. And then they worked we helped them set up their screening, and I was also a consultant, but they really did all of the drug development. They screened and did chemistry for 10 years, made more than 4,000 copies of compounds, and then it took them another 6 years to do clinical and preclinical. And then,
sorry to interrupt, but during that time, how much back and forth was there? Were they just kind of off working, or were they providing insights for you as well?
Early on, there was a lot of back and forth, and then pretty soon it became medicinal chemistry, for which I couldn't make a great contribution. And so it was actually wonderful. Every couple of years they'd say it's working, because early on we were working all together a lot, and later on we weren't. And so it took them 16 years to get approval, and then another 3 years to get approval for prevention. So they deserve enormous credit.
Yeah, it's just a spectacular story. What's one thing that you wish, from the academic side or the biotech side, industry side, you wish that the other side understood better about your work?
Do you want to take that?
Sure, I'll do that. So I'd say the biggest thing is GMP. One of the biggest things, if you're developing a product to take into human clinical trials and doing your own manufacturing, the lab behaviors, documentation, quality management system, how you maintain the lab equipment, is very different from when you work in an academic lab to when you start, going under FDA track, under GMP for human use at a larger scale. And it's quite a learning process. It was a big learning process for our team, implementing, a very comprehensive quality management system, and then how you have to maintain all the equipment and classification of foods in there. It's a big learning experience and a costly journey, but it's a necessary one, depending on how far you yourself want to take a therapy versus going out to a CDMO, for example.
Maybe I'd flip that question around a little bit. The thing I think I learned more from Gilead than vice versa. I mean, obviously we help them with capsid, but these are spectacular scientists, and you learn about the level of rigor in industry. When we published our paper together, their numbers for reproducibility, we have like n equals 3, and we struggle to get there. Their numbers were like n equals 847. And then sort of the level at which they know the literature, because there's a focus. I think I learned an enormous amount from them. It's not that I didn't think there were good scientists going in, but I was just continuously impressed at how much they knew, how they kept innovating.
That really resonates with me. My interactions with industry, the quality of the science and the quality of the scientists has really blown me away consistently. I think that's sometimes a misperception in academia. Well, I would say we're at a fairly unique moment in time, both for biotech and for academia, with a lot of uncertainty. Would each of you comment briefly on what you think we need to be doing to ensure that this uncertainty doesn't stall out innovation?
Yeah, I think, Randy, one of the biggest things all on everyone's mind is we hear about the changes, Health Human Services and the FDA. The unfortunate reality is that the regulators are a big partner of ours in getting these therapeutics through the pipeline in there. They can make or break you. They have a huge impact on the timeline and your cash burn when you're a therapy developer in there. So the thing I basically say is you want to really structure your submissions to the FDA with very well-structured data, very factual and well-written, and you want to be truthful with them and develop productive relationships. I've seen organizations that have gotten confrontational and it doesn't help. The thing I think we have to realize is that it's an education process. I mean, what we've experienced over the last 3 years, and it took us 16 months to get our phase 3 IND approved, is that they had a lot of remote reviewers. You didn't always have the same reviewers looking at different IND amendments you'd submit, so you have to re-educate them there, and you get on calls and have to be very patient in reminding them of stuff and keep things moving and to be persistent but be positive and once again not confrontational. But that's a big thing, is understanding the regulatory pathway and what you're going to deal with. It'll be frustrating, but if you do your job as far as how you structure information and develop more of a partnership relationship, it can be more helpful.
So the FDA has major layoffs. How's that going to affect the process?
I think anyone who's submitting IND amendments or even a BLA right now, it can certainly affect in there. I mean, there's been major disruptions. Dr. Marks left, as well as Dr. Vergan and a number of people in the CBER branch that my field deals with in there. So Macquarie is making a lot of changes yet, so it will have an impact for people right now. We were fortunate to get a lot of this review done before all that hit, but it's going to take a good year for a lot of this to clear up. So once again, find out who your review team is, make sure you're providing very good submissions, take advantage of accelerated designations. If you can take advantage, if you apply for something like fast track or RMAT designation, there now is what's called a coupon program they have. It's for accelerated BLA review. If you're 2 years out from your BLA submission, they're starting that program this year. So you want to take advantage of any of these opportunistic programs.
Thanks. So the question was changes in sort of the funding structure, basically. And as a department chair, I'm genuinely worried. So the proposals are 40% cut in NIH funding and fourfold cut in indirects, which is money that we really rely on, and even worse in NSF and some of the DoD researchers. And so I actually think everybody needs to understand this would be devastating for the University of Utah research enterprise and for the biochemistry department. We have all these talented young people, and I'm genuinely worried. So I think one thing I would ask those of you in the biotech sector, we need allies, and we need it sounds self-serving when we say increase NIH funding. So, but this is the pipeline for new workers. This is the pipeline for innovative ideas. And so I would ask you guys to I'm not allowed to speak for the university, I'm told, whenever I say something political. But if you care about this, contact our representatives, our delegates. Especially Senator Curtis, it's going to come down to a few moderate Republican votes. And so this is really important. Otherwise, it's going to change the way we do business. So that's one thing. And then personally, I'm very concerned about PEPFAR. So that's the George W. Bush program. So the U.S. has taken this enormous lead in supplying HIV drugs and clinical support to the developing world. And it's a George W. Bush program, and the numbers are really staggering. It's saved 26 million people. There are 6 million babies that were born not HIV positive that would otherwise have been. And it's been folded into the U.S. It's in USAID, which has been folded into the State Department and cut badly. But just this afternoon, the Senate, this rescission process, deleted the clawback from PEPFAR. And so I think that tells you that there are moderate Republicans who really care about this, and we should all care about this. This is humanity. So I actually think we're at an existential time, and we shouldn't say we're not.
Yeah. Well, thank you for that. Well, we're going to just wrap up with a little rapid fire session. You get one or a few words to respond to each of these questions, okay? And then we'll get back to socializing.
Thanks for warning us.
Yeah. All right, question one. One biotech or science trend that you're excited about or skeptical about?
Ooh, excited or skeptical. Getting to approval in the next 5 years. And
That's a trend?
At a reasonable reimbursement to recover our development costs.
Okay. Wes, something you're skeptical about or excited about?
I won't say AI, that's too easy. I think what I will say is that I think the pressures on academia and maybe the pressures on the industry could have a catalytic effect in bringing us together. I mean, you look at what China's doing, and it's actually this amazing academic industry partnership, at least as one reads about it. And I think we can do this better, and so maybe that's exciting.
Okay. Next question. A word or a few words. The best or worst career advice you ever got?
That'd be fun.
Getting into a regenerative medicine startup late in your career. So I'm not that it was the best advice, but I think it'll end up being a blessing in the long term. So choose carefully.
My thesis advisor was Steve Lippert, a very distinguished chemist. And he said to me once, Wes, you have to figure out how to make a better first impression. So I've never been able to do that, but now every time I meet somebody, I think, why am I not making a good first impression?
We are making a good impression here tonight. A scientist or thinker who influenced you deeply?
I'll take that one. I was a postdoc with Aaron Klug, who won the Nobel Prize alone, which is an unusual event. And he was just one of the, I've had the privilege of being around all kinds of amazing people, but he was sort of on a different plane, how he thought about structure and function and integrated biology and physics.
So,
Your scientist or thinker?
Actually, I'll talk about one that I was close with, Larry Kukikoff, who was our scientific founder in there. He's now deceased, but he had a way of visualizing cell biology and how cells work together and participated. And he had a Russian accent. He wasn't real eloquent in articulating how it worked, but he described it in simplistic but understandable terms. And that was during the research phase. And darned if it didn't prove out to actually have that mechanism of action once we did all of our preclinical work.
awesome.
And finally, if you weren't in science or biotech, what would you be doing?
Yeah, it's hard to say because I haven't been in that area ever, so I don't know. Maybe a DJ or something. But I'm glad I stayed in that.
I couldn't hit the curveball. Otherwise, I'd be a baseball player. I love baseball.
Awesome. Well, please join me in thanking Bob and Wes for their contributions to the world.
Thank you. Questions?